Hello everyone, welcome to my channel Biochemistry by Dr. Tejas Shah. Today we will see about the overview of amino acid synthesis and degradation under protein metabolism lecture series. So first we see about the synthesis of amino acids. So plants and microorganisms can synthesize 20 amino acids, but mammals only synthesize 11 non-essential amino acids and the rest 9 essential amino acids. As the name suggests, essential means they must be provided in the diet. So carbon skeletons comes from the intermediates of the important pathways like glycolysis, TCA cycle and HM shunt, also called pentose phosphate pathways, and the amino group in the form of nitrogen derived from the nitrogen fixation, which is ultimately derived from the glutamate and the glutamine. The regulation of synthesis of amino acids is mainly done by the mechanism called feedback inhibition. So when there is sufficient amount of amino acids in our body, so it is ultimately regulated by allosteric mechanism as well as the transcriptional inhibition. So collectively it is called feedback regulation. So however, amino acid levels are always maintained at a level such that the amino acid tRNA synthetase can remain active and protein synthesis can continue. So these are some of the important points of synthesis. Now some of the important biosynthetic families of 20 amino acids which are mainly derived from the main glycolytic pathways. So first, from the TCA cycle, the intermediate is the alpha ketoglutarate, which forms a glutamic acid. Further, glutamic acid helps to synthesize glutamine, proline and arginine amino acids. So all are collectively under the glutamate family. Next intermediate is the oxaloacetic acid, which forms the aspartic acid, and aspartic acid further gives rise to the formation of asparagine, methionine, threonine and lysine. Threonine also helps to synthesize isoleucine. So all are collectively under the aspartate family. Next is the glycolytic pathway, in which the intermediate is the 3-phosphoglycerate, which helps to form the serine, and serine further synthesize the cysteine and the glycine. So all three are the serine families. Next is the pyruvate intermediate of the glycolytic pathway, which forms alanine, valine and leucine. So they are under the pyruvate family. Then from glycolysis and the pentose phosphate pathway, the intermediate from glycolytic phosphoenol pyruvate plus erythrose 4-phosphate from the pentose phosphate pathway helps to synthesize phenylalanine, tyrosine and tryptophan. All are the aromatic family. And further, the ribose 5-phosphate from the HM shunt pathway, which helps to form the histidine. So these all are the biosynthetic family of amino acids. In that, some are the essential amino acids and non-essential amino acids. Now the degradation of amino acids. So the site of the degradation is the liver, the major site, but few specific amino acid degradation occurs in the muscle and in the kidney. So first step is the removal of the alpha amino group from the amino acid, that is in the form of process transamination and transdeamination that we have already discussed. You can also go through the video on the same transamination and transdeamination reaction. Then the deaminated amino acids are enters or converged in the glycolysis or the TCA cycle as intermediates. Okay, so this we have already seen. Proteins from the diet and endogenous proteins, digestion and absorption will lead to the amino acids. So the first step in the amino acid catabolism is the removal of the ammonia in the form of removal of the amino group and removal of the alpha keto acid, that is the carbon skeletons, which forms the ketone bodies, glucose and the carbon dioxide. So collectively in the summarization, if we see, as amino acids are degraded, their carbons are converted to first carbon dioxide, compounds that produce the glucose in the liver, that is pyruvate, and the TCA cycle intermediates like alpha ketoglutarate, succinyl coenzyme A, fumarate, and oxaloacetate. And third is the ketone bodies or their precursor acetoacetate and acetyl coenzyme A. So the amino acids which carbon skeletons form glucose in the liver, they are called glucogenic amino acids, and the carbon skeletons which forms the ketone bodies, they are called the ketogenic amino acids. Few amino acids carbon skeletal form both glucose and ketone bodies, and those amino acids are termed as glucogenic as well as ketogenic amino acids. So these are the conversion point of the amino acids. Various amino acids under catabolism, they enters into the main oxidative pathway, that is glycolysis and TCA cycle. Now here one point is to be noted that phenylalanine tyrosine can form acetoacetyl coenzyme A and acetyl coenzyme A, and that is helpful for synthesis of ketone bodies. Here phenylalanine tyrosine also form the fumarate, which enters into the TCA cycle and also help to form the glucose. So they are termed as glucogenic as well as ketogenic amino acids. Now some important coenzymes which also helpful in the synthesis and degradation of amino acids. First is the pyridoxal phosphate, which helps in the transamination reaction under the catabolism and the donation of the amino group for synthesis of various amino acids. Next is the tetrahydrofolate or the folic acids, which help in the one carbon metabolism and also helpful for synthesis of glycine and degradation of serine and histidine. Next is the tetrahydrobiopterin. It helpful in the hydroxylation of the phenylalanine to form the tyrosine. And fourth is the cobalamin, that is vitamin B12, which helpful in the one carbon metabolism, that is in turn transfer of the methyl group. So this is all about the overview of synthesis and degradation of amino acids. Thank you everyone. Like, share and subscribe to my channel. Click the bell button to get all the notifications. Thank you all.